Thank you so much. Thank you to the organizers for having us here. This is a great forum, a great opportunity to open our minds to a more wide frame than our specific work. The title was proposed by the organizers, and I left it. So I'd like to start again reminding what has been said, that humans co-evolve with their microbes, that are specifically adapted to occupy human niches, human body sites, environments. And that these microbes play crucial roles in functions of the intestine, nutrition, immunity, a lot of interaction through the gut barrier, the gut functions, and also the brain with a whole new area in the microbiota studies that have to do with brain-gut interactions. And again, as has been said repeatedly this morning, we are changing our lifestyles, and we have done so at a very fast pace in the recent times. And from a diversity of traditional lifestyles that humans develop and culturally evolve during human history, to a single lifestyle, regardless of the geography. We are living a very common lifestyle that exerts important impacts, not only in the environment, but also on our human physiology and human microbes. And as it has been shown already, what is occurring is a disappearance of infectious diseases, but a rocketing of chronic diseases that have to do with ill immune responses, bad education of the immune system. These diseases are going to be the challenge of the future of health systems. This shows per capita health costs in 36 countries. The burden of these diseases will be unbearable, will be unsustainable, and it's going to be a big challenge. Why are these diseases rocketing? We certainly don't know for sure, and there are two hypotheses. The first one that was proposed is the hygiene hypothesis, that what it states is that it's the altered environmental exposure to microbes that increase the risks of asthma and allergies. A second hypothesis is the disappearing microbiota hypothesis. That is really the lack of our own human co-evolved microbes what disrupts the regulatory pathways, including immunity, in early life. I'm going to show you evidence for both. I think probably both are important. And in relation to the environment first, we are also, as Luis, studying urbanization gradients. I study them in South America. And we chose a gradient at the same latitude in the Amazon basin, Amazon River basin, from hunter-gatherer communities in the jungles, in the forest of the border of Peru and Ecuador. Going east in the Amazon River basin, you have communities that are rural with incipient urbanization and transculturation, to rural towns like Iquitos, and then you arrive in the city of Manaus. And with this gradient, social classes also emerge that were not present in the traditional communities. And in this setting, we studied microbes from the houses and from the inhabitants of the houses, architectural, environmental parameters, microbiome, and metabolites. So in a paper that we already published that is called Walls Talk, we show how the walls of the houses, the microbes that are in the walls of houses, really discriminate between the communities, from the jungle community in red to the rural communities and Iquitos and Manaus, the urban settings. And basically, the reason for these environmental changes in microbes in the homes of people is an increase in human skin bacteria and reduction in soil bacteria. The reasons are urban design, architectural design, and habits. We further analyze mass spectrometry. So these are metabolites in the same gradient from the red Checherta jungle community to the green rural, blue Iquitos, and yellow and orange Manaus. We can see first a clustering apart of the metabolites in the surfaces of the house between urban and rural, but even within the rural, we can observe a gradient from the rainforest to the more rural Iquitos. We can see the gradient in the bacteria component of the surfaces of the houses, and this depicts the diversity of metabolites on top of the bacterial map of the bacterial principal coordinate analysis. And what this shows is that the diversity of metabolites increase with urbanization. Metabolites were less diverse in the forest than in the houses of Manaus and Iquitos. And we see that a little bit here. Metabolite diversity increases, and another microbe that increases is the diversity of fungi with urbanization. So on this other hypothesis, the microbiota, we have been studying the microbiota from traditional cultures mostly in Venezuela, where we find south of Venezuela and north of Brazil have a lot of villages with tribal peoples of Amerindians. And we had described, in collaboration with Jeff Gordon originally, how the US microbiota, this is the digestive microbiota, was reduced in diversity compared with Malawi or Guajibo. So it doesn't depend on geography or ethnicity, but it seemed to depend on lifestyle. And a more recent access to samples from first contact Yanomamis by a team of medical doctors in Venezuela, and we had access to the samples, show that their diversity is much higher than the transculturated Amerindians. So there is a clear tendency to decrease gut diversity with urbanization. We can tell what bacteria are disappearing that are overrepresented in the green Yanomami, and those include some bacteroidetes from the S24/7 group, the sulfovibrio, helicobacters, oxalobacters, prevotellas. And we can see really clearly that there are two big clouds in the principal coordinate analysis of the microbial communities, where the US really cluster in a different cloud, and we can see where the Amerindians cluster in the non-urban or less urban cloud. This is probably the most important slide of my talk. This is from a recent paper we published on the Hadza microbiota. And what we did was take the microbiotas of the gut that have been published in the world and order them in the principal coordinate 1. And what happened was a spontaneous ordination by position in principal coordinate 1. And what happened was the ordination by urban level from jungle savanna populations that occupy the left area of the principal coordinate 1. with the Yanomami in one extreme, going through rural peoples and urban peoples in this cloud, including Japan and China, which we still see that are eating more traditional, less processed diets, but they clearly belong, their microbiota belong to the urban microbiota. The same Italy with their Mediterranean diet. So it's really, you are urban or intermediate or forest savanna type of microbiome. Not only the gut microbiome is changing, decreasing diversity, we are also studying the vaginal microbiota in Amerindians, in part because Amerindians have an extremely unacceptable high rates of death due to cervical cancer caused by HPVs. We have a project in a narrower gradient of urbanization in Venezuela, really going from deep in the forest, same ethnic group, going into the capital of the Amazon state in Venezuela, which is really a rural town, Puerto Ayacucho, and Daniela Vargas did her PhD project studying microbiota diversity and also HPV diversity. And in this principal coordinate analysis, we show the Amerindian microbiota, vaginal microbiota in red, segregating from the mestizos. But if you if you separate the Amerindians from medium, high urban level, low, medium, or high urban level, you see the gradient of the structure of the vaginal community moving towards the space occupied by the mestizo bacterial community structure. The diversity in this case is higher in Amerindians that are in the high urban setting. The diversity from mestizos was the highest. So there is an association of increased diversity in the vagina. So the vagina, unlike the gut, is an ecosystem of naturally low diversity. And perturbations will tend to increase the diversity, whereas in the gut, perturbations tend to decrease the diversity. We found a lot of unique types of bacteria in the low urban level Amerindians that we are characterizing. We can see visually the increasing diversity in mestizos and high urban level Amerindians in relation to the low. Cervical human papillomavirus, which is the cause of cervical cancer, the prevalence of HPV, human papillomavirus, was very high, similarly high across the gradient, 78% of the people. But the Amerindians, when we look at infections by only high risk, which is the purple, only low risk, which is the pink, or both, which is the gold, what we see is that Amerindians, regardless the gradient, tend to be colonized or infected by human papillomaviruses of only high risk group, in the group of the highly virulent. Whereas in the mestizos, these infections were more similar in prevalence. We can see in this graph also the gradient, the increased tendency of HPV diversity with urbanization, with the mestizos having the highest diversity. But that diversity contains equally high and low risk HPVs. Furthermore, we look at the urine of these women, and this is untargeted metabolites, mass spectrometry. When we look at the metabolites in the urine by proportion of traditional diet that they consume, growing from 100% traditional diet to very low in red, you see a gradient in the metabolites. We can look at the same data with looking at the low, medium, or high exposure Amerindians, respectively in red, blue, or orange, and the mestizos. So the metabolites in the urine also correlate with the urbanization level. So women are not the same. These are most of the human microbiome projects that are being done in today's science are studying these hosts, not these hosts. So I'm very pleased to see that there are such a good groups working in the Pasteur and in Europe on this field. It's very hard to get funding to study these populations and to study health. We have learned from ecologists, from forest ecologists and from marine ecologists, in this case, Robert Payne, that if you perturb an ecosystem that is mature, it recovers. We call that resilience. If you perturb an ecosystem and before it recovers, you perturb again, extinctions occur. If you perturb an ecosystem that is not quite mature, it leads to extinctions. So we have compounded perturbations. We do practices in our urban lifestyle. We do a lot of, we have habits and practices that are heavily antimicrobial, that impair transmission or colonization or survival of microbes. We do a lot of intrapartum antibiotics, even during vaginal delivery. In the US, any woman who has strep B will have antibiotics during labor. We do a lot of C-sections, 33% in the US, over 90% in Rio de Janeiro, over 80% in Rome. In South America, it's a very interesting sociological question. It's a behavioral, there is a lot of C-sections by choice. Then we use a lot of preventive antibiotics, such as the ophthalmic ointment, which is the practice in the US. Any baby will have this ointment in the eyes, even if the baby is born by C-section, which doesn't make sense, since this practice was developed and recommended to avoid the baby to get gonorrhea and chlamydia. Then we use formula feeding and further antibiotics and then processed food. So I work on C-sections, and the diseases, there are diseases that have been epidemiologically associated with increase of, with C-sections, and those are asthma, type 1 diabetes, celiac disease, and obesity. These are part of the same diseases that are rocketing in urban societies. How does C-section affect the microbiota acquired by babies? In this principal coordinate analysis, this is our work we did in the Amazon in Puerto Ayacucho. We are showing the microbial communities of the mouth of the mother, of the vaginas of the mothers in red, and of the skin of the mother in dark purple. Any side of the newborn, and these are right at birth, we swab the babies at birth, and if babies were born vaginally, any body side of the baby during the first 24 hours, because the meconium takes a little longer, will be similar in the bacterial composition to vaginal. And babies born by C-section have bacteria in their different body sites that resemble skin. And we think this skin is coming mostly from the environment of the operating room, shed by other patients, the cleaning team, nurses. This is a metagenomic view of the vagina of a mother perinatally. And what we see is a lot of firmicutes and proteobacteria. Visible bacteroidetes, mainly Bacteroides, visible actinobacteria, mainly different bifidobacteria and propionibacterium. How does the baby gut select from this first inoculum? What the baby gut does in 2 days, this is a 2-day-old baby, really at the expense of the firmicutes, the baby has expanded bacteroidetes and actinobacteria, mostly Bacteroides and bifidos. A C-section born, these are pool of 4 babies because we are doing experiments in mice. A kid born by C-section, 2-day-old baby, really has expanded proteobacteria, still has firmicutes, no expansion of Bacteroides, and actinobacteria. In collaboration with Marty Blazer, we follow the babies born by C-section or vaginally, and we can tell the curves of the position on principal coordinate analysis of the red C-section born or the blue vaginally born remain segregated, at least during 20 months. And Bacteroides is one of the bacteria that is affected. It's really low, at least during the first year of life of C-section born. They don't catch up. And interestingly, there's a phenomenon that arises late and in the phylogenetic diversity. The diversity of bacteria catches up in babies born by C-section very early, but after 10 months, just by looking at the phylogenetic diversity, you are able to tell again which baby was born by C-section in a very clear way. Babies born by C-section stop gaining diversity at the same rate than babies born vaginally do after 10 months of life. So we are microbial ecologists. The first thing you think of to solve a problem of ecological impact is restoration. So we say let's restore babies that are born by C-section that come from pregnancies that are healthy. Mothers are not infected, we checked, and mostly we end up selecting, recruiting mothers that had C-section due to malposition presentation or to previous C-sections. We incubate a gauze in the maternal vagina before the C-section procedure, kept the gauze in a sterile container, the 15 to 20 minutes that takes the surgeon to take the baby out, and then swab the baby starting from the mouth, face, and rest of the body. And we showed in a paper published in 2016, what are the, in this pilot, it's a small study, what are the bacteria that we could restore, and it included anal Bacteroides and Lactobacillus, and skin Lactobacillus and S24/7. We are still working in the big paper that follows babies up to a year. It became viral to seed babies, and it has caused the warning of the ACOG, the American Association of Gynecologists and Obstetrics, because there is fear of transmission of infections. And still, as they rightly say, we need to know whether this restores health. So is really C-section plus restoring the vaginal bacteria going to protect against the diseases associated with increased risk when C-section birth occurs? So causation. We know epidemiological associations. C-section increases the risk of being obese, for example, at age 7 in 46%. But we wanted to know causation. So Keith Martinez, my PhD student, did study of C-sections in mice, and he compared C-section born babies with vaginally born babies. The C-section babies had to be fostered because we couldn't do survival C-sections. C-sections were performed without antibiotics and were terminal. He followed from birth to 15 weeks the early microbiota and then also the developmental body mass. And we found a phenotype. So animals that are born by C-section grow much bigger. And the effect is already present at weaning. So the effect is really starting during lactation. The effect is bigger in females, but significant in both genders. Because we were forced, when we saw this, we published it, but we really went back and said, well, we need the controls, because C-sections were fostered. So we need a vaginal foster, and we restore. So we decided to do both the control of foster and the restoration of the C-section. And what we are sending to publication as I'm speaking shows that already at 3 weeks, C-section in red, mice born by C-section grow bigger again, that fostering has little or no effect in males or females, and that restoring, which is the orange group, normalizes the growth up to 9 weeks, which is young adults in mice, and then leads to an intermediate phenotype in females and males. So the restoration, the protection or normalization of the phenotype is partial. When we look at the microbiota of these mice, from vaginal delivery in dark blue to C-section in red, and we have foster in clear blue and restoration, the restore group and the foster group cluster together, not quite clusters somehow in between. They seem a little perturbed, but not significantly. And even after 6 weeks, really the outlier group continues to be the C-section. We did intestinal gene expression at 1 week. These are very young mice. And this is the profile of the genes that are overexpressed in vaginal delivery groups. So this is genes that are differentially regulated between the groups. In relation to the vaginal delivery pattern, C-section really switches off the expression of a bunch of genes. And vaginal foster, half of the animals had a normal pattern, but the other half already seemed to be perturbed, seemed to be more similar to the C-section. Restoration, almost consistently, although in less strongly than in C-section, it's already showing a pattern that is altered. So gene expression does not seem to be restored by restoring microbes. This is gene expression in 1-week-old mice. So the health is sustained over the pillar of the host physiology, which is affected by this other genome that we carry on us. So the interaction is what sustains health, and chemical or non-chemical stressors that affect the microbiota may impair physiology from very early in life and really lead to disease. There is a lot of evidence of the microbiota being behind some of diseases, perturbation of the microbiota. There is increasing evidence. And what we are interested is really in understanding what have we lost and can we restore? What are the functions of these lost microbes and that are associated with risk, and can we restore and confer protection? So anthropology is the study of humankind, and obviously it should include the microbiome, which has co-evolved with humans and are a non-human component of humans. So microbial anthropology is the study of development, evolution, and ecology of humans and their microbes. What can we do to reverse the trend? And basically the response is respect our biology. But we don't want to go back to the jungle, obviously. We can be more natural, maybe by naturally sustaining early development. So instead of creating, the leading concept is having spaces for expressing milk, where the mothers express milk, store it, freeze it, and then give it to babies in bottles. I think there is a lot missing there that is natural and probably important. So we are starting studies where we compare babies directly breastfed by their mothers, and that's simply creating family rooms in the workspace where the mothers are, instead of expressing the milk. And this is one of the challenges for women, and one of the underground reasons why women are not reaching the top positions in society. So we need to address this, and there are microbiome issues there, which are relevant to the baby health. Of course, we have to stop using C-sections when unnecessary, but we need to improve birth and avoid pain during labor, live in greener cities, in greener environment, but most important, because we are already perturbed, we have no option than restoring. So we need to really know what to restore and what is the right diet to keep that colonization. Learning what are the associations between microbiota disease, what are the protecting microbes, and understand their evolution, and preserve them. So we need to create a vault of the high diversity microbiotas from peoples that still have them in the world, that are rapidly integrating to urban societies because everybody wants medicine and technology. So we need to make an effort that I think is urgent to create a vault for the future of health of humanity. An inspiration of a real successful case is the Global Seed Vault in Svalbard. And this will necessitate a big team effort from a lot of people globally. I want to thank people in my lab and wonderful collaborators, and thank you so much for your attention.